Good morning, ladies and viewers at home. This is another lecture on our educational series of Omega Bio-Edutainment. And this morning we are considering um, gene expression and gene regulation under microbial genetics. Why I'm waiting behind the camera is just to allow a kind of review to what we have done in the past, and by the time we get to what we intend to do today, I am going to come off behind the camera. The last time we met, we have actually started this lecture on gene expression, where I told us what do we actually mean if we say gene expression. So, and I told you that there are certain genes that are so essential that microorganism tends to use from time to time. And that was when I made an analogy to the theory of use and disuse according to Jean Lamarck. Don't forget, as you have it on the board, what we actually mean by gene expression is the process whereby the information that is encoded in DNA is transcribed into functional proteins. That of how functional genes are being expressed. Apart from that, in the earlier video, we actually talked about the structure of DNA and the components of DNA. I told us that a gene is a segment of DNA, you need to know that is able to make instruction, specific instruction, so as to produce a gene at the end of the day, just a segment of DNA. Then we know by now that chromatin is the combination of DNA which is being packaged or packed with a protein called histones. Apart from that, I also said that the process of gene expression depends on whether we are talking of being eukaryotic or prokaryotic. And the process of gene expression is going to start with transcription. That is DNA. The first step of gene expression is transcription, where an RNA copy of the gene is being made to undergo 4 different processes. Going to be initiation. Initiation is going to be transcription factor, which I told you different types F2, and we have like that depending on the organism we are considering. Either it is prokaryotic or eukaryotic. So initiation is when the transcription. So initiation is when the transcription factor and the RNA, 2 of them, bind to promoter region of the gene of a transcription, and immediately after that, the DNA will start to unwind locally. So the unwinding of the DNA will now stage of elongation, and at that point in time of elongation, that is when the RNA polymerase will synthesize uracil to replace thymine in the case of RNA. So that one will now lead to the last stage of a termination. So it is at the termination that the RNA polymerase must have gotten into a terminator sequence. And I gave you examples of the set of codon that are used as a stop codon, or what you call a termination. Okay, yeah, and that is where that is going to transcription is going to end. That is when the messenger mRNA will be released, and once it is released, it is going to enter the first stage, which is going to be packaging. That is a post-transcriptional modification. So at post-transcriptional modification, especially in eukaryotes, there's going to be 5' capping at 5' end, and which involve the use of addition of a polyadenylation, that is addition of many adenine. So that one is going to be followed with splicing, that is removal of non-coding regions from a coding region. So, and this is a typical example of the distinction between transcription in or gene expression in prokaryotic and eukaryotic organisms. Okay, so the next thing is, after the splicing, the next thing is that the cell is going to enter into a stage of a synthesis of a protein. And at that point in time, when there is going to be synthesis of protein, that is the process that is called translation. So translation also proceeds from messenger RNA, and it is going to be processed into functional protein. That is that, and that one also is going to progress in 4 different stages. It's going to progress in 4 different stages, which is also the initiation, elongation, termination. Post translational modification will also take place, and at that point in time, that is when we are going to have glycosylation. Glycosylation will also take place in that area. After glycosylation, there's going to be phosphorylation, and this process of translation will actually end with cleavage. And after that cleavage, that is when we have production of a functional gene that is being produced. So that will now mark the end of a gene expression. So the next thing after gene expression process is to see how gene expression is being, or gene synthesis is being regulated. And just like I said in the earlier video, that once gene regulation, if gene is not regulated, it is going to lead to a lot of things, wastage of energy, and the process will also feel unsafe. And that was where I actually related the process of gene regulation to us having a very good car, very expensive, luxurious car for that matter, and at the end of the day, you just discover that the car you have bought with several billions of naira only have acceleration, and there is no brakes there. So you just discover money you must have spent on that, and the energy that you must have spent on that is going to be a waste. So that is a gene regulation. So now, what do we actually mean if we say gene are being regulated? Gene regulation involves a kind of subset of processes that involves controlling when there is going to be gene expression, when, where is it going to take place, and how much of this gene is going to be expressed. So we need to know this. And different types of genes actually have their own method of controlling it. And just like I said, in case of a car, the car does not actually have to wait until it gets to the final destination before it is going to apply brake. Along the line, whenever there is any obstacles, brake will be applied. Or even if there is no obstacle, it is not too safe for anybody to continue at the same velocity from the beginning to the end. So the same thing that also happens in the case of gene regulation. So that is why at every point in time, there are different ways. That genes can be regulated. The first one is there is often gene regulation at promoter and enhancer region. So don't forget that a promoter region is an initiator, is a point where the transfer RNA and where the RNA polymerase and the transcriptional factors will bind. It binds on that region. That is when the process of gene activation or gene expression will begin. So at that point in time, and once the RNA and transcriptional factors now move away from the promoter region, that is automatically going to control it. Is it not that even if you continue to accelerate on the same velocity, and if at a point in time there is no more fuel that is going to get circulated into the engine, so that is telling you that whether you like it or not, that car is still going to stop along the line. So the same thing, we have transcriptional regulation. That is when transcriptional factors and RNA polymerase is going to bind to promoter region, and when it binds to that promoter region, it has the ability to activate or repress the process depending every point in time. So enough gene has been expressed, that means those and RNA polymerase bind to promoter region. Instead of it to initiate the process of initiation, it is going to repress the activities of that. Then we also have another section or another point in the transcriptional factors. So that is we have some proteins that serve as enhancers. So there is possibility that certain region of DNA will serve as enhancers. That is, once the RNA and transcriptional factors bind at that point in time, it will tend to promote the process of transcription. But even at that point in time, once the TF, that is transcriptional factors, refuses to bind any longer, you see that automatically the process will come under regulation. So then, apart from promoter and activities of promoters and enhancers, so we also have transcriptional factors. Those transcriptional factors that I told you that they are of different types, that you can have them as a TF, TF3. Each of them, they have their own functions as well. Some of them will serve as anywhere they bind to, they will activate the process of that are silencer. That is, they will to undergo a kind of silent mode. You put them on silent mode. It's as if the cell is going to enter a process of active. Okay, so that is that. Then at a point in time again, at every point in time again, you just discover that there are some transcriptional factors that will also serve as repressors as well, depending on whatever that is actually needed at that point in time. So that is that. Now again, the other level at which protein genes can also be regulated is at the point of post-transcriptional effect. So that is where you have and there are three types of events that is going to take place under post-transcriptional events. Number one is there's going to be alternative splicing, that is alternation in cutting, removing of non-coding region from a coding region. Alternative, this is a pre-messenger RNA effect that is going to be splitted to produce matured messenger RNA that's going to lead to production of different proteins from a single gene. Okay, so the next one is methylase stability, post-transcriptional process. So just like I said, you see, the process at which every gene regulates, is a gene expression varies. It varies. So let's now start from the other section. So from number 1 to number 6 is what we have actually done before. So we are going to start with what happens as number 6, that is number 6, which is post-transcriptional regulation. Happen is a alternative splicing. So this really after transcription is going to make sure that there is production of different proteins that will arrive from